So this is one particular example of a brain donation that we received. We work very closely with the medical examiner's office here in Houston, the Harris County Institute of Forensic Sciences. We have a very close collaboration with them, and that is where we go every morning, and we call the family members of the individuals who unfortunately passed away the night before, and we ask for brain donations. And once we get consent from the family, the medical examiners will obtain the brain for us, and we bring it immediately to the lab. And then we immediately process it so that we can preserve the brain tissue for then being able to look at the cell level and at the genomic level in the brain. In one particular area, we have the entire brain, and we really want to understand how the brain works and how the different regions of the brain are altered in the different disorders. We have been very interested in the dorsolateral prefrontal cortex, which you can see over here. This is the frontal part of our brain that regulates our executive function, impulsivity, behaviors that we all have that are important for our function, but that sometimes can be dysregulated, and they're important for all psychiatric disorders. So we have been working very closely with the dorsolateral prefrontal cortex. We also, for example, for substance use disorders, want to understand the mechanisms of addiction. So for that, we are interested in the basal ganglion nucleus accumbens region. Also, to understand problems with cognition and cognitive performance, the hippocampus is an area that is really important. So eventually we want to look at all these different brain regions and understand how the neurocircuitry is talking to each other in the brain. One of the things that we do with the brain tissue, once we have a particular brain region that we're interested in, for example, the dorsolateral prefrontal cortex, we will go in there, and what we do is analyze the cells at the genomic level. So we do all levels of analysis. We want to understand how the DNA is then made into RNA. So for that, we do transcriptomics analysis. Then the RNA is made into protein. For that, we do proteomic analysis. And what we want to know is what genes are up or down, what genes are dysregulated in patients with the disorder compared to controls. Our controls are individuals that do not have a psychiatric history. We interview the family members to really understand the history of the patient. And we compare cases to controls at all genomics levels of analysis. And we do a really important bioinformatics analysis that integrates all these networks together so that we can understand how the brain is dysregulated. One of the areas of research that we're really excited about is how we can now have the technology to take blood cells or skin cells from a living patient from whom we know their genetic makeup, we know what mutations the patient has, and we can now take those skin cells and blood cells and reverse their memory and make them into induced pluripotent stem cells and then neurons. And those neurons will have that same genetic makeup that the blood or the skin cells had. And so now we can understand what those mutations are doing to disrupt cell function, and specifically neuronal cell function. So we develop these really beautiful neuronal cultures. We can identify the changes that are occurring in patients with schizophrenia. For example, these are neurons specifically from a patient with schizophrenia, from blood cells from a patient with schizophrenia. We made these neurons, and we can compare them against controls of individuals that did not have schizophrenia, so that we can look at the differences that are occurring at the cell level. So what you can see here is the image of actual skin cells that are coming from a patient from whom we also have the brain. So this person was a donor. We obtained the brain donation, and we also get a skin from that same person. And from these skin cells, who are still alive even after death, we can then now make the induced pluripotent stem cells and then make the neurons that I showed you in the other screen. In addition to the brain, we get the brain donations, and we also get a skin biopsy from the same donor. And from the skin biopsy, we get skin cells that are living skin cells. We can make the induced pluripotent stem cell, and from there get our brain models, or virtual brain models, from the same person from which we have the brain. And because it's the same genetic makeup in the brain models as in the brain, we can now compare our models to the brain from the same person. And this is going to be a really powerful approach for us to be able to understand the mechanisms of cell dysfunction that we see in the human postmortem brain. But in the human postmortem brain, the cells are dead, so we cannot really work with the cells. But with these living brain models, we can. And so now we have the technology to compare the two processes.